DEIVASIGAMANI, working as an associate professor and UGC research awardee, she is Marine Biology Department, ANNAMALAI University, Parangipettai. Today I am going to deliver a lecture on fish non-specific defense mechanism. What is mean by defense mechanism? You know very well about that. Any foreign substances entering the body, our system going to react to engulf the foreign substances, whether it is any particles or pathogens. So that way we are going to talk about the especially the fish immune system. There are two categories based on the function wise, that means non-specific humoral defense mechanism and cellular defense mechanism. The non-specific humoral defense mechanism means in case of teleus fishes have been shown to possess non-specific humoral defense substances which are physicochemicals and functionally similar to mammals, but still different features is there. So that way this non-specific humoral defense mechanism namely have some protein substances involved in the humoral defense mechanism. That means antimicrobial polypeptides and complement cytokines, interleukins and chemokines and tumor necrosis factors and interferon and protease inhibitors, lysozyme, natural antibodies and frataxin and transferrin. So these are the components, these are the molecules involved in the humoral defense mechanism in case of fishes. You know, the fishes is a first vertebrate animal. It is involved in the defense mechanism in a different way. This antimicrobial polypeptide is one of the humoral defense mechanism. This polypeptide a small molecule peptide, so which are active against the microorganism. It is defined as a component of this innate immune responses, or the other name known as natural responses, and been found in tissues of the teleus fishes. So these peptide have been found in the not only for the tissues, it is also mucus and liver and gill tissues of the teleus fishes. These component, this molecule, it is a low molecular weight polypeptides, have the ability to break down cell wall of the both gram positive and gram negative bacteria, and also some of the evolutionary conserved and canyon the bacterial polypeptides have been found. And next, humoral defense component, you know that complement system. The complement system means the particles or the molecules involved in the humoral defense mechanism aspect. This complement is generally exhibit the highest active between the 15 degrees Celsius and 25 degrees Celsius and can remain active at temperature at low as -2 -4 degrees Celsius, which is contrasts to mammalian complement system with an optimal temperature of 37 degrees Celsius. It can be activated in three ways, namely classical pathway, an alternative pathway and lectin pathway. In case of classical pathway means which is triggering by antibody binding to the cell surface, but can also be activated by proteins such as ligand bound C-reactive proteins, or directly involved by viruses, bacteria and viral infected cell. So this method is called a classical pathway. And next pathway means alternative pathway which is independent of antibodies and is activated directly by foreign microorganism. And next pathway means it is a lectin pathway which is activated by binding of a protein complex and consisting of mannose and mannan binding lectin in bacterial cell wall. And next component involved in the humoral defense mechanism in case of fishes, cytokines. The cytokine means it is a gas code of pro-inflammatory cytokines is related as a part of non-specific innate immune responses. The innate non-specific responses is very well developed in case of fishes. So, that way in major drawback in identifying the fish cytokines is a low sequences identified and compared to their mammalian counterparts. The low sequences indeed also is a limit the detection of the protein of fish and cytokines by using the antibodies of human cytokines. In general, however, the fish appear to possess a repertoire of cytokines similar to that of the mammals. You know, the cytokines is a tiny molecules. It is involved in the as well as the non-specific and specific humoral defense mechanism in case of the fishes. For example, tumor necrosis factors and interleukin 1 and 2, so many already identify the cytokine in fishes. And next component involved in the non-specific humoral defense mechanism in case of fishes like the tumor necrosis factor, that means TNF. The TNF alpha and TNF beta are the important activators of macrophages leading to increased respiratory activity, phagocytosis and nitric oxide productions. And next component interleukin, the interleukin is present in the tallest fish species are involved in the regulation of immunity through the situation of the T cells. You know, the T cells and B cells, the important cells is involved in the defense mechanism functions. For especially the T cells, it is a major role for humoral defense mechanism in case of the fishes. And going to have the next component, it chemokines. This is an important component for immune defense mechanisms. It is a superfamily of cytokines. This cytokines, it is plays an important key role for movement of immune effector cells to site of infection, whether it is any region is infected in the fish pot or fish region, this cytokines it's going to move and identify the infected areas. So, this compound is a major role for non-specific defense mechanism in case of the fishes. And next component interferon, this is another one important component. This is also the two type of interferon is there in case of the fishes. The interferon alpha and interferon B are the cytokine with a non-specific antiviral functions, that is based on the inhibitions of the nucleic acids replications within the infected cells. This interferon is a major role for defense against the viral infection in the vertebrate host cell. And next component protease inhibitors. So, this is another important component. It is present in the serum and other body fluid of the fishes and particularly in the teleost fishes. The main function of the protease inhibitors is the maintaining by body fluid homeostasis. In case of the fishes, it is very important aspect is going to the balancing the animal whenever we are moving to the one region to another region. That means it's a migratory aspect, the migratory activities. The animal going to migrate to the salt water to brackish water or in fresh water. So, that time this particular protein is involved in the defense mechanism function, and as well as it is also is a main role for homeostasis functions. and it is also involved in the secretion of the proteolytic enzyme. So, this enzyme is also involved in defense mechanism, particularly for humoral defense mechanism function. And next component in lysozyme. The lysozyme is a bacteriolytic enzyme that is widely distributed throughout the body, and it is part of the non-specific defense mechanism in most teleost fishes. And lysozyme has been detected in serum and as well as the secretions, the mucous membrane and the tissue rich in leukocytes, and mainly in kidney and intestine. You know very well about the kidney and intestine is a part of the immune organs. In additional that, having the case of teleost fishes and elasmobranchial fishes, and thymus and spleen, the two important immune organ is important role for defense mechanism and mainly for the humoral defense mechanism. In case of the lysozyme is a very important role for, it is having secretions of the lysozyme, it is to the involved in the humoral defense mechanism, particularly in case of the teleost fishes. And next component is natural antibodies. In teleost fishes, in naturally found is high level of the serum of the fishes, they will provide the immediate and broad protections against the bacteria and viral pathogens, and making these bacteria key component of non-specific immunity. The teleosts are capable of generating a specific immunoglobulin M type of the antibody. It is the first antibody in the fishes that is called natural antibodies. It is important role for, it is kill the viruses and kill the entering the any kind of the pathogens. Immediately this antibody going to react. That means it is called the antigen, natural antibodies against the various antigens. And next component of the humoral defense mechanism, pentoxins. See, the pentoxin means other name known as CRP, that means C-reactive protein. It is found in the serum amyloid proteins, that means SAA, as present in the body fluid of the vertebrates and as well as invertebrates also this component is present. It is commonly associated with the acute phase response of the inflammatory response. So, this component in different stimuli such as tissue damage and trauma or infections and generative various pattern of CRP, that means C-reactive protein. Its production in teleost fishes, for particularly the teleost fishes. So, this component is very important role for defense mechanism, for especially humoral defense mechanism. It is decrease in the serum, that means the negative acute protein, or the level of CRP is increased in serum, that means positive acute phase protein. Although the pentoxin in the teleosts have a recognized role in defense mechanism. An important another component of teleost for a defense mechanism function, transferrin. This transferrin is a iron as a essential element in this establishment of infected by many pathogens, but the availability of the iron in the tissues fluid of the vertebrate is extremely low due to its high affinity for the blood protein transferrin. So, this transferrin is a globular glycoprotein which has high iron solution activities. This protein is a major role for transportation to the protein to one region to another region, and especially for the plant and as well as in animals. In this only the bacteria with a high affinity system for iron absorptions are able to maintain sufficient iron level to grow in the host, and these are the important component in case of the fishes. It is involved in the defense mechanism. In next class you are going to see the what are the cells is involved in the defense mechanism of the fishes. So, I am happy to share with in research area for especially the fish immune system. I am one of the member I am happy to share with my publication when related to the our research area for especially fish immune system in India. I am proud to say our center is one of the center is having very good publication in reputed journal for especially nature publication and scientific report and interview. So this journal, it's very important journal for an academic research institution for especially our Annamalai University is one of the biggest university. I am proud to say having the publication in related to immune system and my research area of human fish immune system. I published this paper, I can share with you, and along with it, I have the patent of my compounded, 2 immunomodulatory compounded and international patent for Australian government and as well as the German patent. I am proud to say and thank you for this opportunity for our authorities of Annamalai University and my dean and director and my good professor and Dr. Saktivel sir. Thank you for the opportunity. Bye. See you next class. Thank you one and all. Thank you very much.